My name is Revati, and I'm a session aide for the 2020 AAAS meeting. Today, thank you everyone for joining us either physically or virtually on a Saturday morning to talk about green toxicology. We are joined by two wonderful panelists, Alexandra and Thomas. So let me start by brief introductions. Oh, I'm sorry, Andre.
Thomas had to go, had to fly already.
Oh, I'm sorry. Okay. Sorry about that. So thank you, Andre and Alexandra. Please, can you introduce yourself and maybe tell us about what you're doing?
Sure. So my name is Alexandra Martins, and I don't think this is on. Okay, now it's on. So my name is Alexandra Martins. I'm junior faculty at Johns Hopkins and the director for the Green Toxicology Initiative at the Center for Alternatives to Animal Testing. And we are basically trying to design assays and to come up with better ways to test chemicals so that we can have safer chemicals with quicker, with less animal testing. Andre?
Yeah, I'm also a junior faculty member at the Center for Alternatives to Animal Testing, Johns Hopkins University, and my main emphasis is more on mass spectrometry and statistics, but I'm also involved in the green toxicology activities.
Okay, thank you very much. So I study chemistry, and I find it interesting how many of the academic labs and industries are engrossed in doing cutting edge research, collecting data. But during the process, when we create molecules or during the drug discovery process, we often forget about the safety aspects. So what are your thoughts on that?
I think that's an important point. You know, one of the reasons that I started working with this, because I had a lot of experience working with research and development chemists, and they always said nobody taught us to think about toxicity. So from their perspective, it's very easy to design a molecule with given parameters. Do I need to make a better sealant, a more persistent dye? Do I need to make a more flexible plastic? If you give them a target, research and development chemists will do it. But nobody told them, oh, and you have to make the molecule look like this, otherwise it's going to be an endocrine disruptor or it's going to be a carcinogen or things like that. So there was very little toxicity training for chemists, and similarly for a lot of toxicologists, there wasn't a lot of training in what the sort of things you need to think about if you're an R&D chemist. So to some extent, green toxicology is really trying to bridge that gap a little bit. The other thing that we thought was important was the more we can front load toxicity testing in the research and development process, the quicker it's going to be to develop better chemicals. So for example, if you want to do a rat test for reproductive and development, that's going to take several years, it's going to take a lot of money, and you have to have already synthesized a lot of the compound. So you can't really do that very early in the process. You can't rule that out before you invest a lot of money in developing that chemical commercially. So we really wanted to develop assays that you could use at the very beginning, so you could fail quickly. If it was a toxic chemical, you could get rid of it. And I think the reality is nobody wants to design a toxic chemical. That's not the goal. Toxicity happens because they really didn't have the right tools to develop a better chemical.
Andre, your thoughts?
Yeah, and toxicity is not what you think it kills you immediately, but also questions like persistency in the environment are pretty important. If you remember DDT, for example, from many years ago, one of the problems is that it sticks around for a very long time in the environment and has very long-term effects. So this is something very important. I also would like to stress out, so one of the ideas, if you are industry and you develop something, a new product or a new ingredient for a product, you normally don't start with one. You start your pipeline with a lot of candidates in parallel. So, and you would like to find out which is your champion, which you would like to advance. And one of the killers in the very end of the development pipeline is that you find out, oh, it's unfortunately a carcinogenic, which means all the money which we invested so far is gone. And it's gone. You can do nothing with it. And so to already take into account that in the very first step in selecting your best candidates to advance them into your pipeline, this is something what green toxicology is also about.
Yeah, I think many of us think of toxicology as disposing of hazardous chemicals or how to handle it, but this topic of green toxicology is something which is new and emerging, and it was very well explained. So how open are the industries in adapting to these new methods of whatever you propose?
I would say industry is fairly open. In many instances, they've been the pioneers. They do not want to put a lot of money into chemicals that turn out to have problems farther down the road. So I would say that industry has kind of been leading the cutting edge in science. I think certainly the pharmaceutical industry, is already significantly advanced, although with, they're working with very different chemicals. But for them, the expense of, getting it wrong clinically is so high that they have much more robust system. In the case of industrial chemicals, I think you're starting to see that more. I think the problem is a lot of the startups that are working in the green chemistry space don't necessarily have, the tools to invest in that. And I think the other problem is that it's great to run all these alternative assays, but when it goes to the regulators, that right now isn't what they want to see. They kind of are expecting to see the standard tests, and all that is changing. That changes at a much slower pace.
Yeah, since you brought about the topic of regulators, what do you think is the role of science policy and communication, since at AAAS we are talking so much about advocacy, science communication, and science policy? What is your message to them?
That's a good question. I think it's kind of a complicated message. I would say that the regulators are certainly catching up. There has been a big push at EPA to switch to animal-free testing, but there isn't necessarily a clear roadmap how we get there, and I think that that's going to be a very complicated policy issues because on the one hand there are people who do not want to see animal tests ever, and on the other hand there are people who just see this as one more way to get out of making sure that chemicals are safe. So there are very different and both valid concerns that are going to have to be addressed and to make sure that everybody feels we have safe chemicals. I think in terms of communication, I think that's going to be a very difficult issue going forward because as it is right now, I don't think that people react realistically to chemical hazard. There's a lot of fear and phobia and misplaced concern about certain chemicals, and I don't know that the switch to in vitro assays is going, or alternative assays is going to make that any easier because they're far more difficult to read and they tend to have a lot of noise. And as Andre can say, with the mass spec, one of the things we've been realizing is that when you can measure things down to the parts per billion, you can discover that we're exposed to a lot of stuff. And people tend to worry about that more, and we're only going to be able to increase our ability to detect these things. So how do we react to that without overreacting?
Yeah, one of the important things to answer your question, I also see in training the regulators, because they are not any longer in science, they are not any longer at the cutting edge of technologies. They are looking at mainly at tests which are many years old, which are pretty static. And so one of our roles at the Center for Alternative Animal Testing is also to go to the regulator and inform them about new techniques and try to make them aware about what's going on, and just give them as an independent broker who has no interest for industry, who is an free academics organization without any conflict of interest, to go to them and provide them with information about these new approaches, new techniques, and so on, that they can think about how to pick it up in their portfolio. And we get now also more and more the feedback that the data is not primarily used from in vitro assays for the registration, but it's more and more showing up in the appendixes to give additional explanations and things like that. And if you're not trained to understand these things, it's difficult for you because you are normally out of the university since 10, 20, 30 years as a regulator, so you cannot, you're not necessarily keeping up with these things. So there, I think, is a role in communication to bring this to bring this information to the regulators, to the people who are making the decisions, and also, of course, to the policymakers.
Yeah, so it seems that most of the research in this area is data driven and you need a lot of data sets. But what my perception is, a publication these days is maybe, I don't know if I can say this, is biased towards positive results. So no one really publishes negative results. If you find out that a molecule is toxic, I am not very sure if it's included in the publication explicitly. So how do you deal with that?
One of the things is the regulatory world is not the publication world. A lot of databases that we are using are not coming from academic research is publishing. development pipelines in industry and regulatory databases, they don't necessarily publish their results in academic journals. So we have to use many different sources of information to develop new assays and so on, and work one-to-one with industry to get data sets and so on, because this data is not necessarily published. Whether it's positive or negative, for both it's the same.
Yeah, I think you bring up a really good point that there is a lot of publication bias, and I think that for certain chemicals they've attracted a disproportionate amount of attention, and I don't think that the publication record on that has served the scientific community very well. To give an example, when we started pulling all the reproductive and developmental toxicants from ECHO, which is just a large regulatory database, there were very few positives. Whereas if you look in the publication literature, there's an enormous number of articles claiming that various different chemicals cause reproductive and development. Now, some of that is different assays, but a lot of that is just publication bias. And you also get this issue when everybody is publishing on a chemical. For example, there are over 10,000 publications just on bisphenol A, and a lot of them have very contradictory things. And at that point, you get such a deluge of data that it's very hard to sort out, what is and is not reproducible and useful from the perspective of a regulator.
So since you work with alternative animal testing studies, there is a lot of ambivalency, as you mentioned, among scientists as well as the general public. Whether is it reliable? So how do you think a study which is done in an in vitro or in vivo alternate model will extrapolate to human bodies too? Because that is the kind of fear or mixed notions that many of them have, that you need to do studies on animals so that it can replicate in humans.
I would say that gives you a false sense of security. I would say that it's very difficult to extrapolate from an in vitro system to a full organism, and it's going to be more difficult for different endpoints. For some endpoints, we can very easily do that. We have pretty good models of skin and eye for corrosion and things like that. We obviously don't have great in vitro models for, other endpoints, right? Neurotoxicity is very difficult for that, although we're making progress with that on the mini brains. But I would say that extrapolating from a rat to a human is also extremely problematic. And a lot of the concerns that people have had about chemicals, for example, developmental neurotoxicity, are especially difficult to see in an animal model. So, for example, with lead neurotoxicity, that doesn't cause, at the levels that most people are exposed to, overt toxicity that you would see in an animal assay. What you do see is a shift in the IQ curve for humans. And that is, extremely problematic, and that would be extremely difficult to pick up with an animal assay.
So please don't forget, the animal assays, they have been defined as valid in the 50s and 60s because there was a need for regulatory testing of hazards and do a proper risk assessment. So they had to come up with something, and they just decided we throw things on animals and we believe in the results. So a lot of these assays have a lot of limitations which you overcome by adding then safety factors to your exposure. And so the reproducibility, for example, of the eye irritation, the Drace test where you put stuff in the rabbit eye, is pretty bad. The carcinogenicity assay, the biorodent assay, is notorically positive. So there are a lot of examples of where the animal test is not really an unbiased gold standard, let's say it like this. On the other hand, the in vitro assays are in this development pipeline. There are several international harmonization and validation bodies. So on European side, the European Center for Validation of Alternative Methods of the European Commission, ICVAM, NICE, ETIM on the US side, and then there's in Japan one, in Brazil one, and then also these things get internationally harmonized and standardized on OECD and ICH level. So there is a very long pipeline that these assays have to walk through to get finally regulatory acceptance. So for example, for skin irritation, which was one of the very earliest replacement methods where was hardly any critic on it, it took until 2010 that the OECD guideline came out, which was an unbelievable long time frame. So these assays are, the people look much more rigorous, right? Yeah, much more rigorous on them to replace the animal test.
And just to kind of, follow that up, the reality is these tests were developed, almost in some cases 100 years ago. So we are using technology that, came around at the same time as the, Ford Model T in the era of the Tesla. And, yeah, there's always going to be problems when you're replacing older tests, but surely we can do better at this point.
Yeah, that's definitely true. Since you're talking about technologies, what are the novel technologies that you're using? Computer modeling, in silico, in vitro?
One of the things that I do a lot at CAD is I use transcriptomic technology to try to figure out what's the mechanism of toxicity. So a lot of times we can do that on a very small animal test, or we can do it on cell tissues. And so it's very complicated because you can't just get out an answer. You have to use fairly sophisticated algorithms to try to tease out the causal pathway from the molecular initiating event to the downstream consequences. But you can do it. And I think that that's proven very powerful to try to zero in on exactly what is the molecular mechanism. A lot of the problems with BPA came because we thought we understood the mechanism, but we really didn't understand it as well as we thought we did. So I think that there's a lot of potential for using those kinds of technologies to get a better molecular readout of what's happening at the cell.
Yeah, on one hand, you have these developments on a mechanism based to understand how toxicity is going on. So not like the black box of an animal. So you just look at the weight of the liver or things like that afterwards, and you don't understand what really happened there. So you look on one hand on the mechanism, and on the other hand, you try to develop proper cell-based models, which means first you need the right source of cells, which means human iPSC cells, for example, or primary human cells with skin, for example. The skin test is done with primary human cells, with foreskin cells, and you have a lot of sources for primary liver cells, for example, which is also pretty important endpoint. And then, so proper cells. Next level is make proper organs out of it, or organoids out of it. So make balls out of it, or print them. That you have the right set of how the test item is entering the organ, and you have the right communication between the cells, because we find out much more now that the cells need to communicate to each other, otherwise they are not acting in the right way as they would do in the human. And then another level above that is then from organ to organism, which is called ADME, absorption, distribution, metabolization, and excretion. Because if you do an in vitro assay in a lab, you have a dose response curve, and you say, I don't know, my 50% effect level is at 1 millimolar. So you go to the toxicologist, and the toxicologist says, how much is that in milligram per kilogram body weight per day? And you say, well, I have no clue. But this is how it's done in reality. So you need to find this translation also from your test tube to the organization, the in vitro, in vivo extrapolation, which especially involves also absorption, distribution, metabolization, and excretion.
Seems like it's quite interdisciplinary. You work with chemistry, biology, and at the interface of data science and statistics. So what are the...
And with policy people.
Oh yes,
Which
policy.
is, without policy support, we would do it in a dark chamber, and it wouldn't help anybody.
So what are the challenges that you face?
I think one of the challenges that's going to be especially important for the new generation of toxicologists and chemists is getting used to working in the era of artificial intelligence. So that wasn't part of my training when I was a toxicologist, but I use data mining all the time, and I think going forward that's going to have to be just a standard tool that everybody comes equipped with. But it's very, very difficult to change the curriculum, and a lot of people, particularly in toxicology, don't necessarily have the math background to totally feel comfortable with it. And I think that that is going to be, something that's going to have to be bridged. And I also think it's important that toxicologists get a little bit more comfortable with chemistry. Most of them, don't have a good background in chemoinformatics, but that's becoming very important. And I think that similarly, chemists need to start getting a little bit more training in the sort of different styles of thinking that are important in the life sciences, so they can kind of understand why toxicology data is a little bit ambiguous, that it's more difficult to reproduce than enzyme catalysis or something like that. So I think that that's what I see as being the important challenge going forward.
In addition to that, I see one of the very big challenges in international regulatory acceptance. So you are not any longer, the US is not any longer an isolated country, European Union is not an isolated country. So if you as a company bring a pesticide on the market, you would like to bring it on several markets which have different regulators. And with the animal assay there is a lot of standardization, so a lot of you can produce the mutual data acceptance from OECD, for example. If you have done your results according to this assay and under good laboratory practice, you can transfer the results from one country to the next. This is much more challenging with in vitro results. So I see one of the challenges I see is in the acceptance of this alternative, in the harmonized acceptance, not individual acceptance, harmonized acceptance, because you cannot do your tox data set for one country one set, for the next country another set, and for the third country a third set. Then you would simply say, well, then I do the original animal assays because it's cheaper. And so this is something I think, but people are working on that. There are internationally harmonizing committees for that, but this is something which takes an extreme long time, because this is, you have to make treaties around these things, and this is something which really takes a long time.
Yeah. So what is your message to academics? So we know industries, you're trying to propose to them to adapt to alternative approaches. What is your message to academic labs or students, whether to integrate this into their curriculum, or what measures can they take to do a safe research?
Oh, that's a complicated question. I don't know where to start. I think that the chemistry curriculum is changing to incorporate more toxicology, and I would say if it's not in your curriculum, definitely seek it out. I always try to point out to chemists that toxicologists are here to be your friends, not your enemies. We're not here to tell you at the end of the line you've developed a bad chemical. We want to work with you to develop better chemicals. So I think that a little bit more focusing on interdisciplinary research very early in your career is important, and I would definitely tell pretty much everybody in any field that you're living in the era of data science, and you should develop a certain, even if it's not going to be the focus of your research, you should develop a certain familiarity and comfort with it.
Yeah, I think to make people aware very early in their career that efficacy of a molecule, so whether it works or not, or whether by changing a property of a molecule it works better than the molecule before, is just one part of the coin. You have to look pretty early on the other side of the coin and think about these questions. And for that you need to understand how metabolization works, and you need to understand that acute toxicity is not all. There are a lot of toxic endpoints and environmental toxicological endpoints which you need to consider. So you should invest some time. You should take a course in to try to understand what is this about, because you cannot simply take 5 new drug candidates and combine them and give them, because then you will very likely have a problem. So it's really something you should think about that as early as possible. It's not about just designing a better drug or a better chemical or a better pesticide. It's also think about what is the flip backside of doing that. And the more early you think about it, the less problems you have later.
Yeah, I think that's a very good point, to think about the safety aspect at a very early stage rather than realizing it later. Yeah. Do we have any questions from the audience at this point? If not, can we have closing remarks from both of you?
I would say that the message of green toxicology is that we can and we absolutely should develop safer, better chemicals that are less toxic, that have better properties. But the way to do that is going to involve different tests and a different way of thinking about how we do R&D chemistry. And it's also going to have to involve a change on the part of the regulators. But these are all things we can do with better science.
I think I can't add anything to that.
So the key is it's a collaborative effort of scientists and the science policy people and communicators to solve this problem of toxicology. Please thank me, and please join me in thanking Alexandra and Andre. And thank you all for physically or virtually joining us today on a Saturday morning. I hope you have a very good time at the rest of your meeting. Thank you.
Thank you.